Students, our next topic of discussion will be cytoskeleton. So these are the skeleton which is present inside the cell. So cytoskeleton. Now the cell requires something to provide its shape, to provide a proper shape to the cell, is not it? So those thin filament-like structures, very thin thread-like, fiber-like structures, they are called as cytoskeletons. There are these cytoskeleton, the main function is to provide cell shape and size. Also, they will help in movement. The cytoplasmic movement that takes place will be due to this cytoskeleton. So, different types of cytoplasmic movements. Then the movements of cilia, flagella, spindle fibers, or the formation of spindle fibers, centriole, cilia, and flagella will take place because of the cytoskeletons. They will help in formation of cilia, flagella, centrioles, and spindle fiber. Now, there are three main types of cytoskeleton, so it can be broadly categorized into three types. First is called as the microtubules. Then the second one, it will be called as the microfilament. And the third one, it is called as the intermediate filament. Now the microtubules, they are composed of tubulin proteins. These tubulin proteins, they will be in the form of dimers. That means two different monomers will be present of tubulin molecule. So dimer of tubulin. So alpha and beta. There are two dimers, alpha and beta. And if we draw it, how it will look? This alpha and beta dimers, they will be present together, and there will be this kind of arrangement will be 13 times in a spiral shape. So it will form spiral shapes like this with 13 dimers around each turn. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So there will be 13 dimers like this, so 2 together. So this kind of arrangement will form the tubulin. Now this is the most thickest of all the other cytoskeletons. So this is the thickest one, you must know. Okay, and it is made up of tubulin protein. Then this helps in the formation of cilia, flagella. This also helps in the formation of sperm tail, and it is also helping in the cells which consume food from outside. These microtubules will be the one which will help in it. For example, in the formation of the pseudopodia and all. Next is the microfilaments. The microfilaments, they are made up of actin protein. The name of the protein which makes it is actin. So there will be two chains of actin polymer. Okay, this is suppose one chain of actin polymer. Here each monomer is one actin, and there will be two chain which is twisted around each other like this. So two chains of actin molecules are present here, and it acts as a cytoskeleton. It also acts as a stress fiber below the plasma membrane. That means it keeps the plasma membrane stretched out. And it will also change the salt to gel state and gel to solution state. Also, cytoplasmic streaming, that means movement inside the cell, will take place due to this microfilament. It also helps in cytokinesis when the cell divides. So during that time, the division of the cytoplasm will take place due to these microfilaments. And the last one is the intermediate filament, which is formed of bundles of keratin fibers. Keratin fibers or proteins. How do they look like? They will be present in the form of small, cylindrical, long, thread-like structures, and they will be always present in the form of bundles like this. Many of them will form a bundle. And from the bundle, we will see that certain arm-like region will stick out. This is what is observed under the ultra scanning microscope. Okay. So these are the three different types of cytoskeleton. Hope the students are clear about it. Now, after the cytoskeletons, now let us discuss about the centriole. There are two centrioles in an animal cell. The two centrioles are always present in a as if they are perpendicular to each other. For example, this is one centriole, it has a cylindrical appearance, and the other centriole will be present perpendicular to it, like this. So one will be like this, and the other will be like that, over it. Okay. So two centriole we will observe, one above the other. And this structure along with around it we will also find certain ray-like structure. These are small filament, and these are called as the astral rays, and each of these particle will be called a centriole. This is the centriole, and these are the astral rays, and this whole structure will form the centrosphere. And collectively this whole thing will be given a scientific name, which is called as centrosome. So this whole thing along with the fluid inside, so everything together, it will be called as centrosome. So this is present only in the animal cells, and the centrosome will duplicate when the cell wants to divide. So there will be two pairs like this. Now, if you want to study deep about this centriole, how does one single centriole look like? So the centriole will have 9 plus 0 arrangement. It is called as 9 plus 0 arrangement. You will know why once I draw it. Now this arrangement will be due to microtubulin. Arrangement of microtubulin. You already know from cytoskeleton what are tubulins, what are tubulin proteins. So same tubulins, they will form the arrangement. And now there is 9 arrangement like that. So 1, 2, 3, 4, 5, 6, 7, 8, and 9. These microtubules, they will be in the form of trimers. That means there will be 3 monomer of this kind of tubulin, alpha, beta, and gamma. Alpha, beta, and gamma. Alpha, beta, and gamma. And they will be all attached with the help of certain fiber to the center. They will be attached to the center like this with the help of some fibers. And then we will also find that the alpha is joined to the gamma with a separate arm. There will be a long arm which will arise from the alpha, and it will attach to the gamma of the adjacent tubulin. So it will happen with all of them, alpha and gamma is going to connect. So this is how the centriole will look like. And outside this also we will find that there will be certain spokes which are present. Spokes made of proteins are also present around it. Now what is the function of the centriole? It will help in the cell division in the animal cells. Now that you have understood about the centriole, let us discuss about the structure of flagella. Structure of cilia and flagella is very similar to each other. So we will take it up together, cilia and flagella. We are going to discuss about the filament portion of the cilia and flagella. We are not going to talk about the basal body. We are going, or the root of the cilia and flagella. We are going to discuss about the only about the filament portion, the long hair-like portion of the cilia and flagella. So in this one, they will have 9+2 arrangement. 9+2 arrangement of tubulin. So it will be very similar to centrioles. If you have understood centriole, you will also understand flagella. So in the center, it will have two dimers are present, and there will be a cross bridge connecting the uh, two tubulin, and around it there will be a sheath which is covering it. There will be a very thin sheath which will cover it. So these together, it will be called as a central microtubule. This is called as central microtubule, or the structure is dynein. The chemical composition is dynein. There are two, that's why dynein. Two tubulin, so it is called as dynein. Then around it, there will be presence of dimers of tubulin. So dimers of tubulin, and that also nine numbers. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9, and they will be as dimers. So alpha and beta. Suppose this is alpha, this is beta. So alpha, beta. This is alpha, this is beta. So this is the arrangement in flagella and cilia. This are connected with a small filament to a spoke which is very near to the central microtubule. This is attached to the sheath of the central microtubule, and this alpha molecule, the alpha tubulin, it will give out two arms. So it will have one arm like this and another hook-like arm present. So all of them will have two arms. We can draw two arms which is arising from the alpha tubulin. One looks like a hook, another one is erect out. The inner hook and the outer hook. Now the inner arm, it is attached to the beta, and the outer hook, it will just remain suspended. So the inner arm, it will attach itself to beta in all the cases. So all of them, they will attach to beta. So the inner arm is attached to beta and the outer hook will remain suspended like that. Okay. Then around this structure also there will be a sheath which is found. A sheath or a membrane covering will be found. outside the 9+2 arrangement as well. This sheath is very similar to the plasma membrane, or it is exactly like plasma membrane, you can say. So you can also write it as plasma membrane sheath. And these alpha beta, they are tubulins. Just remember that they are tubulin proteins. And what is the function of cilia and flagella? You already know. They help in the movement of the cell. Flagella will be present either 1, 2, or only few in numbers on a cell, and cilia will be present in large numbers all around the surface of the cell. So I hope this is clear to all the students.